Good morning. Good morning, everyone. Yes, if you would, just type your name into the chat for me. Thank you. Thank you very much. Good morning. I hope everybody had a good weekend. Hopefully you had a chance to get outside a little bit. All right. So today we're going to be talking about aseptic technique, which is basically a collection of all the things we do in the microbiology lab in order to achieve our most important goal, which is don't contaminate the samples with ourselves and don't contaminate ourselves with the samples. So it's actually a collection of techniques that we use that we refer to as aseptic technique. This is our busy week. Remember, this is the week that you have your next lecture exam and your first lab practical exam. So very busy week this week. In fact, I think the first thing we'll do this morning is go ahead and pull up our to-do list. See if I can do this. Stop. Let me try one more time. Okay. So hopefully that's what you're seeing on your screen right now, our modules page in our Canvas course. I'm going to scroll down to this week's module. We are on week 4, and it's our first really busy week. So I'm going to click on the to-do list. And here it is. There's both a lecture exam and a lab practical exam, as well as our regular weekly work. We have one lecture topic this week, the acellular microbes. And we have a lecture quiz on that topic due by Wednesday at midnight, just like normal. And then on Thursday morning, the lecture exam opens up and the lab practical exam opens up. And as I said last time, I would recommend that you take your lecture exam first and then take your lab practical exam. The lecture exam covers 3 topics. I've got them highlighted here: prokaryotes, eukaryotes, acellular microbes. And just like last time, the lecture exam will open at 8 a.m. on Thursday morning, and it's due by Saturday at midnight. So you can take it any time between Thursday morning and Saturday night. You just have to find a slot of time that you can sit down and take it. Remember, it has somewhere around 25 to 30 questions on it. You have 90 minutes to complete it once you begin, for full credit anyway. You can, of course, take longer if you need longer, but I don't think anyone will. We have laboratory topics this week. As usual, we have aseptic technique today. We have microbes in the environment on Wednesday. As usual, you'll have a lab homework due by Sunday at midnight. But as I said last week, I really recommend that you get that homework done as soon as possible after Wednesday's lab. You can't do it until Wednesday after we have lab, because there'll be some questions on Wednesday's topic. But I would recommend getting that done Wednesday afternoon, Wednesday evening, if possible, because I will grade those and get them back to you as soon as you submit them, and you'll have that material for studying. Lab practical exam opens on Thursday, just like the lecture exam, but it's due Sunday by midnight. So you have a little extra time for this one. I did make a change on this list. I made a change on this list. It used to say that the lab practical exam will cover all of what we've talked about in lab, but now it's only covering most. There's one topic that we're going to have covered that will not be on the lab practical, and that's the topic we're doing on Wednesday. So let me say that again, because that's important. On Wednesday this week, we're going to talk about microbes in the environment and how we look for them and how we test for them, how we count them, so on. This topic, microbes in the environment, will not be on the lab practical. The lab practical ends with the material we're doing today, aseptic technique. So biosafety, that was our first topic. Care and use of the microscope, examination of living microbes, that was the wet mount and the hanging drop, simple staining, gram staining, and aseptic technique. Microbes in the environment is not on this lab practical. It'll be on the next one. So I'm going to click back here onto modules for a minute and scroll down to our week. Here's your lecture exam link. Remember, you have to use the lockdown browser to get at the exam. And just like the first exam, it's worth 80 points. I have to grade this exam by hand. So the score that you'll see when you submit it is not your final score. And then down here, here's the link for the lab practical exam. Now, there's something very different about the lab practical exams versus the lecture exams. The lab practical exams, like the name suggests, are supposed to be very practical. They're supposed to be about the practical things that we're learning in lab, because really that's what laboratory is about. It's about the practical workings of the microbiology lab. There are 12 questions on this practical, and some of those questions have you looking at an image and answering a question about that image. In fact, several of the questions have you looking at images. Now, if you think about the topics we've covered so far, there have been a lot of images that we've looked at together. For example, in the biosafety lab, we looked at the signage that we put on laboratory doors. And we looked at the microscope and all its pieces and parts in the microscopy lab. We looked at how to make a wet mount, how to make a hanging drop. We looked at slides of bacterial smears that had been stained, simple stains and gram stains. We've looked at a lot of images. So you can expect to see some images on this lab practical exam, and you'll be asked questions about them. Keep that in mind while you're studying. Now, there is a difficulty in Canvas when you want to put a picture into a question or an image. Depending upon what kind of computer the student is using, sometimes those images show up fine, and sometimes they don't. So in order to make sure that everyone can see all the images, what we do on the lab practical is a little bit different. I actually give you a document, a file with all the questions on it and all those images. You'll open up that file and then you'll answer the questions in Canvas. Okay? So let's take a look. When you click on this lab practical exam, when you're ready to take it, this is what you're going to see. It says Download the file below to see the questions for this practical exam. Then type your answers into the Canvas quiz. Now, I have a typo here. It says "eep your answers." It should say "type your answers" into the Canvas quiz. Okay, so what you're going to have to do is first download the questions, open them up in a window on your computer, and then you'll be answering them just like any other quiz. Okay, so you can see when you look at the actual quiz, there's no questions written here. It just says answer question 1 here, answer question 2 here, and so on and so on. Answer question 3 here. So this is where you type your answers in, but the actual questions are on this document. Okay, so you'll have 2 windows open during the exam. One window will be the questions, and one window will essentially be your answer sheet, where you'll be typing in your answers. Does that make sense? Does that make sense to everybody? We had a problem one semester. We had a problem with a lab practical because some students' laptops wouldn't open the pictures in Canvas. When they got into Canvas, all they saw was, a blank where there should have been an image. And so we went to this format instead. So you'll have to open this file and then type your answers into Canvas. So Brooke is asking if that causes a problem in Lockdown Browser. Brooke, what Lockdown Browser is not going to allow you to do is navigate away from Canvas. It's not going to allow you to go to, for example, Google or another website. But this, you'll still be in Canvas here. You'll just be working in 2 different windows in Canvas. Okay, that makes sense? So I've given you a big hint that there are some pictures on this lab practical. There are 12 questions and at least 6 of them have pictures, maybe more. So when you're studying, when you're reviewing the notes and the slides from lab, think about what I might ask. What might I ask you? I can't ask you a whole lot of different questions because you have a lot of different topics, right? I can only ask you maybe 2 questions, 3 questions from each topic. So what questions do you think I might ask you? That's a really good exercise when you're studying. Ask yourself, what is she going to ask me about biosafety? What is she going to ask me about care and use of the light microscope? What exactly is she going to ask me about simple staining? Think about it. Think about what maybe you would ask if you were the instructor and it was your job to test students on their knowledge of these lab topics. So you haven't had a lab practical yet, so I know it's a little nerve-wracking, but try not to be too nervous. You'll have plenty of time. Again, students typically take about a half an hour to finish this lab practical, if that. So don't worry about time. That won't be a problem at all. Okay. And of course, as usual, if you have any questions while you're studying, before you take the practical, you can message me through Canvas. All right. So very busy week ahead. Be sure to get everything in. And if you're not sure whether you've gotten everything in, go to the to-do list. That's what it's there for. The to-do list at the start of every module is there so you don't forget anything. Okay. All right. Okay, any questions about what's going on this week? Many of you have already done your acellular microbes lecture and the acellular microbes quiz. Good for you. Good for you for getting it done. It's a difficult topic because it's so different from all the other topics we've discussed so far. The viruses are a fascinating group of organisms, but boy, are they strange. From the fact that they're not technically alive to the fact that they have to invade a host cell in order to get replicated. They are very, very strange organisms and important to us in microbiology because remember, bacteria and viruses are the two most common types of pathogens in humans. So very important for us to understand them. All right, I'm going to go ahead and pull up our slide set for today about aseptic technique. Hold on just a sec. I'm going to try this again. All right, I'm at the bottom of the slide set. Here we are. So what you should be seeing on your screen right now is the title slide. Lab Aseptic Technique. Our objectives for today are to first define this concept of aseptic technique. We're going to talk about why they are used in the laboratory, why they're so important, and we're going to talk about how to implement these techniques while we're working in the lab. So when you hear someone talk about aseptic technique, when you read, for example, on a protocol, something saying, be sure to use your best aseptic technique. What that refers to is all of the different methods that we use in the laboratory to prevent contamination. Now, remember, there's two kinds of contamination that we're trying to prevent. We want to prevent the contamination of our culture or our sample with ourselves. We don't want to get microbes that are on our bodies into our samples. And we don't want to contaminate ourselves with our culture or our samples. So we don't want to get things that are in the culture onto our bodies. And we have to work very hard to meet those two goals. It's very important to remember that microbes in the laboratory range from perfectly harmless to pathogenic. And because of that, we treat all cultures that we create in the lab as though they are pathogens. Put a star next to that in your notes. Even if you know that you are working with an organism that is not pathogenic, cannot hurt you, even if you know that, you treat that organism as though it was a pathogen. Always. Every single culture that you create in the lab, every single diagnostic sample that comes into the lab, you treat as though it has pathogens in it. If you do that, if you treat every sample as though it's pathogenic, then when a real pathogen does come in, you'll be all set. You'll be all set because you've been using the right techniques all along. Treat all pure microbial cultures as pathogens. And we could also add, treat all patient samples that come into the diagnostic lab as though they were pathogens. If you are carrying a cup of urine that was collected from a patient, if you're carrying that down to the diagnostic lab, you treat it as though it is full of pathogens. And if you're not careful, it could harm you. That's how you treat it. Now, I want to say just a couple things about culture media. Remember, when we use this word culture, what we're talking about is a collection of microbes in some kind of a growth medium that we are purposefully creating in the lab. So a culture is growing microbes. And in the laboratory, we typically grow pure cultures. And what that means is there's one organism growing in there. That's our goal. Sometimes we create a culture because we only have a small number of cells and we need more of them. Maybe we want to do some testing, for example. So we would take those few cells, excuse me, and we would put them into a culture media to allow them to grow. And when I use that word grow, what I really mean is divide, make more and more and more cells. So culture media is kind of interesting. There's all kinds of culture media used in the microbiology lab. Some of it is solid. Some of it is what we call semi-solid. Some of it is liquid. It's basically a material that contains nutrients and water and other components that the microbe needs in order to grow. We say that culture media gets inoculated with microbes. All that word means is that we are adding microbial cells to it. We are inoculating that container with cells in order to encourage those cells to grow. We take our inoculated media and we incubate it over some period of time. Now, a lot of times when we hear this word incubate, we think about heat. We think about an incubator, right? We think about putting the container into this box that keeps a warm environment for us. But incubation is really more about time. So yes, sometimes we incubate in an incubator, but other times we incubate in room air. There are some organisms that we work with in the microbiology lab that will grow best for us at room temperature. For example, things that live out in the environment often prefer room temperature. So we can keep our culture containers in a dark area at room temperature. Sometimes, though, especially if we're dealing with organisms that come from the human body, we need to give those organisms more warmth. If you think about it, if it's a bacterium, a pathogen, for example, that grows inside of us, think about the streptococcus organism that causes strep throat. That's a bacterium that has evolved to grow well at our body temperature, 98.6 approximately. So when we want to grow that streptococcus in the laboratory, we need to put it into 98.6 degrees. So it will grow for us. That's what those box incubators are for. We can set the temperature and give that microbe its ideal conditions for growing. Most microbes like to be in relatively warm, dark, and relatively moist conditions. So incubators are often also humidified, so there's plenty of water. So one of the most important things for us to remember is that growth media, and we're going to talk more about the different kinds of media in a future lab, but understand that the kind of culture media that is designed to allow microbes to grow, that medium will grow both our desired microbe and any other microbe that gets into that container. So we have to be very, very conscious of that. We are literally creating an environment that encourages microbes to grow. when we make an agar plate or a broth tube. So yes, we're going to put some cells in there. We want them to grow. But if a contaminating microbe gets in there, that's going to grow too. So we have to be very careful to protect these containers from getting contaminated. Now, here's some general rules for us. Number one, never leave a culture container open any longer than absolutely necessary. That includes an agar plate, and we'll look at some of those today, a broth tube, or any culture media container. We never leave them open any longer than is absolutely necessary. Remember, microbes tend to fall. They don't jump up off of surfaces. They tend to fall out of the air. So we want to keep our containers covered as much as possible. Now, obviously, sometimes you have to open them if you want to put cells in or you want to take cells out. But you want to do that quickly so that you can cover that container again and reduce the chances of something falling into it. Number two, always assume that surfaces are contaminated. So we never place sterile materials on a surface, a work surface, even if we just disinfected that surface. If you've got a sterile material in your hand, you never put it down, because if you do put it down, you have to assume it's now contaminated. Number three, remember that microbes fall, they don't fly up. They don't jump up off of surfaces and get into containers. They fall from the air down into our containers. Nearly all forms of contamination are actually being carried around the lab on dust particles. And those dust particles are constantly swirling around. We can't see them, they're microscopic. But that's where the source of contamination is. Contaminants fall onto or into sterile surfaces if we're not careful, if we don't handle things correctly. Now, are there any questions about those general rules? We're going to take a look at, again, the handling techniques today. We'll look at a video together in just a few minutes. But does anybody have any questions so far? What I want to talk about next is a little bit about waste disposal. Now, we've already talked about waste disposal in terms of stains. We said that all of the stains we use in the laboratory are considered hazardous. And so we have to collect all of our waste stain and put it into a bottle that's marked as hazardous waste. We also talked about the autoclave very briefly. We said that when we have materials like culture containers that have microbes growing in them, when we're finished with them, we have to kill those microbes before we dispose of them. And that's what the autoclave is for. We put those materials that are ready for disposal, we put them into the autoclave, and the autoclave sterilizes them with heat and pressure. Once they're autoclaved, everything is dead. So we can toss that material into the trash at that point. So we've talked about those things, but there are a couple of other really important things for us to understand about how you throw things away in the lab. Remember, we use a lot of microscope slides in the laboratory, microscope slides with cover slips sometimes. Those things are single-use items. We use plastic, unfortunately. We use plastic, sometimes plastic test tubes. There are plastic inoculating loops that you can buy. We use tips on our micropipetters to move certain volumes of material around. All that stuff has to be disposed of. And the question is, how do we do it correctly? So that's what we're going to talk about next. This is a very busy slide. Too busy, really, but I wanted to put all of this information down for you so you weren't scrambling to write it down. This is a list of different things, just generally how they're disposed of. So some of the things we use in lab are considered hazardous waste. So we keep containers in the lab to hold that hazardous waste, generally used stains and any other chemicals that we might be using. We pour that material into these hazardous waste containers. We label those, we put them in a safe place, and a registered hazardous waste disposal company comes and picks those up for us so they can be safely decontaminated and disposed of. We also keep a regular trash can in the lab. There's a lot of things that can just go right into the regular trash can. All of our gloves go into regular trash. So even if you think you might have gotten some bacteria onto a glove, you're still free to put the glove in the trash. Paper towels that you might use, they go in the trash. Little Kim wipes. Kim wipes are just very small, essentially pieces of paper towel. Lens paper that we use to clean the lenses on our microscope. All of this stuff can go into the regular trash. Now you might ask yourself, wait a minute, how come I can throw my gloves away if I've been handling microbes? What if there are bugs? What if there are bacteria on my gloves? How can I just put those in the trash? Isn't that a danger? The answer is, it's a danger just in that moment, but within minutes, that danger will be gone. And that's because your gloves are dry. They're just dry material. They have no nutrients. They have no water. So even if you got some cells onto your gloves, those cells have no way to survive because there's nothing there to help them survive. The same is true of paper towel, for example. When you disinfect your desktop with paper towel, even if you picked up a few cells on that paper towel, there's no way for those cells to survive in the trash can, and they'll be dead within minutes. So those kinds of dry goods that we use in the lab go right into the regular trash. Now, you'll notice there's a star next to sharps containers because there are actually two kinds of sharps containers that I want us to be familiar with. I'm sure you all have seen sharps containers in your life and perhaps in your work. These are commercially available containers that have very particular signage on them. They are a very noticeable red color. And it's a place for us in healthcare settings and laboratory settings to deposit sharp things that might cut someone if we put them into the regular trash. Now, what I want you to notice is that there are actually two kinds of sharps containers that you might come in contact with in a laboratory. In a doctor's office or a hospital setting, the only kind that you're going to come in contact with is a contaminated sharps container. So a contaminated sharps container contains things that have come into direct contact with either bacterial cultures or body fluids. These are the kinds of sharps containers that most people are used to seeing, and we'll take a look at these in just a minute. If it's touched bacterial culture in the laboratory, or if it's touched body fluids from a patient, we have to assume that it has pathogens on it. Remember, we assume that microbes are pathogenic. So we have to put it in a particular sharps container that will be treated as though it has dangerous microbes in it. What you may not have seen, though, are other types of sharps containers that contain sharp things, but things that have not come in contact with any microbes. So we'll take a look at these two kinds of containers, and we'll talk about why we have both kinds in a laboratory setting anyway. There are special containers for glass waste in the laboratory. We'll take a look at that. And we'll take a look at the special bags that we use to put things in for autoclaving. Inoculated culture plates that we're done with, disposable culture test tubes, anything disposable really that comes in contact with pure culture, we throw into an autoclave bag and we put through the autoclave. Now I've got a note down at the bottom that's important. If it's a glass test tube or a glass flask that we are using as a culture container, those are reusable. So what we have to do is get all the culture material out of that glass container into an autoclave bag. Then we wash the glass container and then we autoclave it empty. Let me say that again, because that's important. If you've got a plastic disposable test tube and you've got some broth in there and you've been growing microbes in there, when you're done with that, that whole container will go into an autoclave bag. It'll go into the autoclave. It will be put under heat and pressure, and all the cells inside that plastic test tube will be killed. That whole thing will then be put into the regular trash and sent to the landfill. But let's say you're in a laboratory that's using glass test tubes instead of plastic. Glass test tubes can be used over and over and over again. So how do we manage those? Well, you wouldn't put the whole tube into the autoclave bag. You would empty the culture into the autoclave bag. And then you would take the tube and wash it. You'd wash it with soap and water, and we have special laboratory soaps for that. And then you would take those empty glass test tubes and you would put them through the autoclave, not so you can throw them away, but so you can sterilize them. Autoclaves do two things for us. They make trash safe for disposal by sterilizing it. And they also make containers and instruments and certain garments sterile so that we can use them over and over and over again. So sometimes what we're putting in an autoclave, we're trying to sterilize it so we can use it again. Other times, the things we put in the autoclave, we're trying to sterilize so we can throw it out. So we actually have two very different uses for our autoclaves. In a hospital setting, a lot of what goes in an autoclave is going to get used again. Surgical instruments, surgical gowns, containers, medical supplies that have to be sterile. A lot of things get autoclaved so that they can be used sterile. But other times what we're putting in the autoclave, we're getting it ready for disposal. We're trying to kill any microbes that might be on it or in it so that we can throw it out safely. Now, when it comes to aseptic technique, remember, it's really a collection of techniques, and some of these we've already talked about. We talked about how to decontaminate your work surface. Remember, we said there are two types of disinfectants that are used most often in a laboratory. Those are 60 to 90% alcohol and 10% bleach. Either one of those is perfectly good for disinfecting your workspace. We also talked about how we have to properly organize materials. We have to keep our personal materials away from the workspace. And we have to have a nice open area where we can set everything up and get our work done. We've also talked about one of the tools we use quite often, which is the inoculating loop. Remember, it has an aluminum handle, and then it has a wire coming out of one end with a loop on it. And we use that loop to move cells from one container to another. We said that you have to sterilize that loop. You have to sterilize it before you use it, so you don't accidentally contaminate your culture. And you have to sterilize it after you use it, so that you don't accidentally contaminate something else. So we always sterilize that loop just before we use it and just after we use it. It's the wire portion, remember, the loop and the wire, that we have to get red hot, for example, inside of our incinerator, or red hot inside the flame of a Bunsen burner. Once metal glows red like that, you can be assured that it is now sterilized. Remember too, we talked about how important it is that you let your inoculating loop cool off for a little bit before you stick it into a culture container. Because if you put a hot loop into, say, a tube of broth, you're going to kill any cells that are right in that area. So let it cool off for about 20 seconds before you put it in. So we've talked about some of the techniques, but there are some we haven't discussed yet, and that's what we'll be talking about today. We're going to talk about a technique known as flaming. I'm going to show you how we flame a glass test tube. We're going to look at how we aseptically transfer bacteria from one container to another. So for example, from a broth culture that we've been growing into a new sterile broth tube. If you remember, we said there's a technique called subculturing that we use in the lab. If we grow cells in a test tube for a day or so, we can then take some cells out of that container and put it into some fresh broth, a new container, to allow our cells to continue growing without having a problem of running out of nutrients or having too much waste material build up in the tube. So we have to learn how to subculture, how to take cells out of one tube and move them into another tube, or how to move cells from a broth tube onto an agar plate. You can also move cells from one agar plate to another agar plate. It's most of what we do in the lab is actually move cells around from one place to another. We have to do it using aseptic technique. We'll talk about agar plates today, specifically how we handle those, because they require a little bit of special handling. We'll talk about some of the other tools we use when we prepare cultures. And again, we'll look at Sharps containers and autoclave bags in terms of waste disposal. So this is the Sharps container that most of you have probably seen. These are present all over any healthcare type of environment. So you'll see these in a doctor's office. You'll see these in hospitals. You'll see these in patient rooms in the hospital. You'll see them in diagnostic laboratories. These are commercial Sharps containers. And the idea is that any sharp object that you are done using, you drop through the slot into the container. When this container is full, one of those specialized commercial companies will come and collect it in order to put it into the autoclave and sterilize everything that's in here. It also then gets disposed of safely because, again, there are sharp things in here. So you wouldn't want those things just to go straight into a garbage can or a trash can, because someone might get injured, someone might cut themselves with that material. So they come in all shapes and sizes. You have to buy these from a medical supplier. You'll notice that they all have a label on them that looks like this one over here. They all have this universal symbol on them for biohazard. And that's because not only are the things in a sharps container sharp, but they're also contaminated. In the healthcare setting, these are the kinds of things that touch body fluids. Needles, for example, the needles that come with syringes, those needles are used to take blood samples or to withdraw fluid from certain places on the body. Those kinds of things belong in a sharps container when you're done. They're contaminated with those body fluids. Scalpel blades and things like that go into sharps containers. Once they're full, they're treated as biological hazardous waste. So that's an important distinction for you to be aware of. When it comes to dangerous waste material, there are actually two different categories. There are what's considered hazardous waste. Stains, for example, are hazardous waste. And there's what's considered biohazardous waste. Biohazardous waste is things that could make you sick, like microbes. So both kinds of waste have to be handled by certified commercial companies in terms of disposal. But you might guess, and you'd be correct, handling biological hazardous waste is actually more expensive than handling ordinary hazardous waste. So we separate the two kinds of waste. Biohazardous waste is any waste that contains microbes or potential microbes that could infect a person and make them sick. Regular hazardous waste is chemical waste. Both of them are dangerous, hazardous. Both of them have to be disposed of by certified commercial handlers. It costs money for those commercial companies to haul your waste away and take care of it for you. But biological waste is more expensive to dispose of than regular hazardous waste. That's why we keep them separate. So in some laboratories, not in doctors' offices or anything like that, or hospitals, but in some laboratories, you'll find two kinds of sharps containers, regular sharps and contaminated sharps. The sharps container, the regular sharps container, is for sharp objects that you're disposing of that have not come in contact with any kind of bacteria or any kind of cultures or body fluids. Just sharp things. The contaminated sharps container has sharp things that have come in contact with culture material or body fluids. So two kinds of containers you'll see in a laboratory setting. And a lot of people will say, why? Why bother doing that? Well, it's all about money. It's all about money. It's about costs. So take a look at this slide. Up at the top, what you're looking at are some non-contaminated sharps containers. And yes, we recycle things to use as sharps containers. The only rule about this is that whatever container you use as a non-contaminated sharps, it has to be opaque. In other words, you can't see through it. And it has to be hard plastic. And you have to have a cover for it. Okay, it has to be opaque. You can't be able to see through it. It has to be hard plastic. And it has to have a cover. And what we do is we just cut a little hole in the cover, a little slit in the cover, and we write on the container that it contains sharps, non-contaminated sharps. So old laundry detergent bottles, those are real commonly used. This is an old coffee container. People who work in laboratories save these kinds of bottles at home and bring them in and donate them as sharps containers. This one over here, you would not be able to use. This is just a milk jug. A milk jug would not be an appropriate sharps container because it's opaque. It's not opaque, sorry. You can see through an empty milk jug. This one over here would also not be an appropriate sharps container because it's not hard material. The sharp things could poke through a plastic bag. So you can't use a plastic bag to hold sharps. So any one of these over here, you could use as a non-contaminated sharps. And then of course, for your contaminated sharps, you would use one of these. It has the biohazard symbol on it. It's ready to receive biohazardous material. The reason we have two kinds of sharps containers in some labs is because it saves us money. It's less expensive to dispose of sharp things that have not been contaminated. You can take a non-contaminated sharps container straight to the landfill. Now, you shouldn't put it in your trash can. You should carry it to the landfill because it's got sharp things in it. Generally, you hand it to someone at the landfill and you tell them, hey, there are some razor blades in here, or there are some scalpel blades in here, or there are some non-contaminated needles and syringes in here, and they will dispose of them correctly. But generally speaking, you don't have to pay for that, except in your taxes, I guess. So if you have one of those red containers that has the biological hazard symbol on it, you can't just take that to the landfill. The landfill won't take that because those have contaminated sharps in them. Those have to be picked up by a commercial handler and disposed of correctly. So it saves money to manage your contaminated sharps separately from your non-contaminated sharps. I actually keep a sharps container. I keep one in my home just because I'm so trained on this. And anytime I have an old razor blade, or sometimes I use a jigsaw. If I have an old jigsaw blade, I put it right in my sharps container because I don't want to put that in the trash because it might harm somebody. I'm very well trained. Now, we use an awful lot of microscope slides in the laboratory, and even though they're made out of glass, they are considered single-use items. Remember, most of the time we are heat fixing cells onto those slides. So it's not something we use over and over again. Glass slides are not thrown into ordinary trash, even though the cells are dead. Remember, once you heat fix and stain a bacterial smear, the cells are dead. So it's not that they are a source of contamination, but they're glass. And microscope slides break really easily. So we always dispose of them in what are called glass trash containers. These are just cardboard boxes. You can buy them from any laboratory supply house. They're about 3-4 feet tall, maybe a foot or two wide. And what goes in here is glass that has been used in the laboratory that we're ready to dispose of. These things are not considered contaminated, like I said, because the cells are dead when they're on a microscope slide. Now, one note to put in your notes, one thing to write in your notes that's not on that slide. Remember, a depression slide that we use to make hanging drops, a depression slide is considered reusable. So when you're finished making a hanging drop, what you do is you wash that slide and you use it over and over and over again. And the reason is, well, we're not heat fixing anything. into a depression slide. So there's nothing stuck in there. And they're expensive. Depression slides are commercially made with that concave well in them. And they're expensive. They're, $10, $15 a pop. So we don't throw those away. We reuse depression slides. But regular microscope slides, regular cover slips, those get thrown into that glass waste box when we're done with those. So this waste is not considered contaminated. It's just sharp, and that's why it's dangerous. All right. Does anybody have any questions about anything I've talked about so far? Again, this is less about aseptic technique than it is about just being a good steward of the environment, being a good steward of other people, and a good steward of the environment by throwing your waste away correctly. It is really important. It's on us as people who work in laboratories. It's on us to make sure that we keep the public safe from our material, and we make sure that we're not contaminating the environment in any way. All right, this is a good point for us to take a break. It's just about 5 of 10. Go ahead and take a short break now, get yourself a cup of coffee, whatever you need to do. And I'll see you a couple minutes after 10. Swabs.
Hey, Professor.
Yes.
I have a quick question. It's kind of random.
That's all right.
It's actually from the lecture on acellular microbes.
Yes.
So this part where they said a virus can infect a bacteria, but then I was thinking, and this might be obvious, can a bacteria be infected by a virus and then become pathogenic to a human? Like, you know what I mean?
So the kind of viruses that infect bacteria cannot cause disease in us.
Okay. Because I was like, can you get a double infection?
Yeah. Actually, it's kind of fascinating because there are some people now who are trying to think ways that we could use bacterial viruses to deliver things into our body, because they don't harm us.
Okay.
So every living thing, every cellular organism on this planet has a virus or more that can infect it, but they are evolved for that organism.
Okay.
So the bacterial viruses, they don't harm us, which is kind of fascinating, right?
Yeah. Okay, cool.
That's a good question. That's a good question. There are also viruses that infect archaea.
Yeah.
There are some viruses that infect plants. There are some that infect animals. Yeah.
Okay. All right. Cool.
They are very cool. They are very cool things, viruses, but unfortunately they cause a lot of misery too. All right. So we're just about ready to start again. If you would, go ahead and type your name into chat for me. Thank you very much. And we'll get started again. Yeah, the other thing to remember about viruses is, just like with the other microbes, there are some viruses that are completely harmless. There are viruses that are part of your microbiome. That always freaks people out when you tell them that. There are viruses that live on you and in you that don't bother you at all. And then there are pathogenic viruses. So we have to change the way we think, especially in the general public, about microbes, because it's a very small percentage of microbial organisms that actually harm us. Most of what we encounter doesn't hurt us at all, and in fact helps us in some ways. Yeah, I always freak people out when I talk about that. When I talk about their microbiome, they get a little weirded out. All right, so I'm going to jump in again now into the slides. We have a video to watch together, which is going to demonstrate the techniques that I mentioned earlier that we need to know, the aseptic techniques. So remember, we said we need to know how to flame a glass test tube. We're going to take a look at that. We need to know how to move cells from one container to another and do that aseptically. And we need to know how to handle agar plates. Agar plates are a little more cumbersome to handle than broth tubes are. So we have to learn how to do that correctly as well. So that's what we're going to look at in this video. Let me pull that up for us. Let's see if I can do this correctly this time. When we're working in the microbiology lab, hold on. When we're working in the microbiology lab, we are often moving bacterial cells from one place to another from one media to another. And there are different reasons that we do this, but we always have to use our very best aseptic technique while we're transferring cells so that we don't cause contamination to occur. Now, there are lots of reasons why we would take cells out of one culture container and move them to another. One of the most common reasons we do this is because cultures get old. Remember, when we put cells into, for example, a broth culture, there's a certain limited number of nutrient molecules in there. And as the bacterial cells start to grow and divide, they're going to use up those available nutrients. They're also going to be excreting waste materials into that culture tube. And that's going to be dangerous to the survival of the bacterial cells. So from time to time, if we want to keep a culture going, we have to take some cells out of the old culture and move them into fresh media so that they can begin to grow and divide, again, with plenty of nutrients and plenty of space. We call that process subculturing. When you take cells out of an existing culture and you move them into fresh media, we call that subculturing. Now, as you've seen, there are really two primary types of culture containers that we encounter most often in the laboratory. There are test tubes, and those can have liquid media in them. They can have a semi-solid media in them. They can even have solid agar in them. The other container we see is the Petri dish, the plastic single-use lidded dish. We often refer to these as plates. And those agar plates have a thin layer of solid media on the bottom. So those are the two containers that we encounter the most, and those are the two containers that we move cells between most often. So what we'll look at in this video is how to do all of that moving using our very best aseptic technique. Set up in the workspace here, there's an incinerator that's been plugged in and is nice and hot. And there's an inoculating loop. The inoculating loop must be sterile because you would never lay a contaminated inoculating loop on the desktop like this. The only other piece of equipment that I'm going to need to do this demonstration is a Bunsen burner. I don't have access to a Bunsen burner today, so I'm going to use something as a substitute, just for demonstration purposes. And that something is a bottle of hot sauce. That seems appropriate, a good substitute for a Bunsen burner. Now, the next thing I need is a broth tube. And I want you to notice that it's a glass tube. We've talked about how to open up any test tube with culture in it by holding it at an angle and by holding on to the top of the tube in our fingers. What we need to remember is that the inside of a sterile tube of uninoculated broth is still sterile until we place something into it. The outside of the tube, though, once you have touched it, it is no longer sterile. Now, when we're working with a glass test tube, we can take an extra step to protect the sterility inside that nutrient broth tube. And that's to flame the mouth of the test tube when we open it, before we handle it, before we put culture material in, and also flame it again before we close it. Now I'm going to show you what I mean by flame it. This is something we can do using aseptic technique to add an extra layer of protection against contamination. People don't always flame glass test tubes. Sometimes it's just too time-consuming of a procedure, but I want to show it to you because it is, again, an extra layer of protection against accidental contamination. You would never do this with a plastic test tube, though, of course, because you would melt the plastic. So the test tube that I'm holding in my hand has a liquid broth in it, a nutrient media, and bacterial cells growing in it. And I'm going to take a sample of those cells out of this tube, and I'm going to flame the mouth of the tube before and after I remove those cells. So you'll notice that I'm going to take the cap off. I'm going to hold the tube at an angle, just like always. But here's the extra step. I'm going to pass the neck of that tube, the mouth of that tube, through that flame. Let's take a look at that again. Remember, the bottle of hot sauce is my Bunsen burner, and we're pretending there's a flame coming out of that black cap. So I'm going to take the neck, the open part, the mouth, if you will, of that test tube, and I'm simply going to pass it through the flame. Now, what does that achieve for me? The opening to the test tube is the place that's most vulnerable to a contaminating microbe falling in. My hands are up near the mouth of that tube when I'm taking the cap off. And of course, I'm also a source of contaminating microbes, even with gloves on. So by flaming that opening to the tube, what I'm doing is I'm applying heat directly to the glass. So if there's any microbes sort of right on the lip of that opening, I'm killing those. I'm also briefly warming up the air that's in the mouth of that tube. And by doing that, I actually create a little current, a little swirling current of hot air. And just for a moment or two, that's going to protect the tube. It's going to prevent anything from falling down out of the air and going into the tube. So I've created a few moments of protection from accidental contamination. Now, after I'm finished doing whatever it is I'm going to do with the tube, I'm going to pass it through again before I put the cap back on. Again, if I have accidentally introduced any microbes during my procedure, I just killed them. And I've created a little current of air right at the end, just in case there were any microbes hovering over that area, that opening. I've killed them with that heat. It's a very brief pass through the flame. It's going to kill cells that might be on the lip, the outer lip of that tube that might be a source of contamination. And it's going to create a current of hot air just in the mouth of the tube, just for a couple of seconds, that will keep that entryway free of contaminating microbes. Now we're going to watch a transfer of cells from one tube to another tube. This tube has my bacterial culture in it, okay? And this tube has clean nutrient broth in it. Notice that I put the clean broth tube up higher in my hand. I'm going to widen our view here a little bit. Now, the other thing I'm going to need is a Bunsen burner. And of course, I don't have one. So I'm going to substitute in my bottle of hot sauce. Okay. Now, first thing I'm going to do is sterilize my inoculating loop. That needs to be into the incinerator until the wire is red hot. For purposes of the demonstration, I'm going to shorten that up a little bit. We'll pretend that we're red hot. Now we have to hold it until it cools off. Remember, approximately 20 seconds. You can count in your head. Now it's important that you get that loop comfortable in your hand for the next part. I've got my tubes at an angle. I'm going to take off both caps. And the reason I do that is because I'm going to be flaming. So I'm going to flame the culture tube first. Now I'm going to take out a sample from the culture tube. I'm going to flame it again. And now I'm ready to close the culture tube. Take one of your caps and put it on. Flame the clean broth tube. Place your sample into your plain broth tube. Swirl it around a little just to make sure it all gets in there. Flame it again. Close it with a cap. Remember, your loop is dirty right here, so don't forget to put it back into the incinerator to sterilize it once again. If you're working in a laboratory that doesn't use open flame, you can still do a successful broth-to-broth transfer using your best aseptic technique. Just going to want to remember to hold both tubes at an angle while they're open. You're going to want to avoid touching the neck or the lip of the tube as much as possible. And you're going to want to close those tubes just as soon as possible to help prevent any accidental contamination. Now let's take a look at the next kind of transfer, which is from a broth tube to an agar plate. Remember, we use plastic single-use Petri dishes in the microbiology lab, and we can pour agar into those dishes and allow it to solidify, giving us a solid media to work with. We refer to those as agar dishes or agar plates. The agar, the solid material, has nutrients added into it so that the bacteria can grow. So this is an agar plate. This happens to have nutrient agar on it. You can just see it. It's a thin sort of beige film on the bottom of the plate. Now I'm taking the lid off just so you can see the inside of the plate. There's just this beigeish sort of sheen to that media. Very, very thin layer of media in here. It feels a lot like gelatin. And it's easy to push your loop into that gelatin. So you have to be careful when you're handling it. Oh, I've mentioned before that we handle agar plates upside down. We put agar plates into the incubator to allow bacterial cells to grow upside down. In other words, we want the agar, that solid media, to be at the very top once it's upside down. And what this does is it allows any condensation that might develop inside that nice warm incubator. It allows it to drip off of the surface of the agar and onto the lid of the plate. This is helpful when we're trying to grow bacteria because that condensation might interfere with the growth of bacterial cells on the agar surface. So we handle them upside down, we open and close them upside down, and we place them into the incubator upside down. I'm going to move my incinerator into place here as I get going. My inoculating loop, my tube full of bacterial culture. Now, this inoculating loop is clean, it's sterile, and I want you to notice that I'm going to start bending this wire. The wire is wire for a reason. When we're inoculating things, especially the surfaces of plates, we can bend this wire to make it easier to spread the cells out. Now you can bend it as far as you want or as little as you want. It really depends on the person handling the loop. But once you bend it into place, you can still get it into the incinerator bent because it's very flexible. It's a wire. So you would heat up the wire just as you always would. See, the bend is still in it. You're going to let it cool. If you have it bent as severely as I do here, once you open up your culture, which I'm doing here, Just going to want to be very careful as you introduce the loop. Again, you don't want to touch the neck of the tube. Take out some of your culture material, close your culture. Now you're ready to go. So I'm going to open this dish. Again, it's upside down. And I'm going to keep the plate at an angle so that I can put these cells on the surface. I'm going to place the loop on the surface, and I'm going to make a nice zigzaggy motion all the way across. I'm going to take my loop, remember, incinerate, sterilize, get rid of those cells. So essentially what I've done is I took a bunch of cells out of the culture tube, the broth, and I placed them onto the first place that I hit on that auger. I put a large number of cells, a large amount of cells onto the surface. And then by sliding my loop across the surface, back and forth, what I'm doing is I'm spreading those cells across the surface of the plate. So that I'm giving them room to grow on that nice nutrient-rich solid media. Let's take a look at that again. Plate comes up, loop touches the media, and then without lifting it, I'm going to just streak it out, streaking it back and forth in a zigzag motion, spreading out those cells. As we go through the summer and we look at other techniques that we use in the microbiology laboratory, we'll also look at different ways that we can streak an agar plate, depending on what we want to achieve in terms of the growth of the cells. Now, anytime you open a container that has growth media in it, nutrient media, or any other kind of media, remember what we said. You want to keep that container open for as short amount of time as possible. You want to quickly get it inoculated with cells, and then you want to close it again because you want to prevent contamination. Now, different people use different techniques to achieve this. You can open and close an agar plate a couple of different ways using aseptic. Here's an agar plate that's being held right side up. And in this technique, we're going to use the lid to keep the plate partially covered. See that? I can reach in between with my loop and do my streaking pattern, close it, and then put it upside down. Let's look at that again. I'm going to keep the plate right side up, use the lid as partial protection from microbes falling onto it, streak it, close it, back upside down. Now let's take a look at a couple of ways that you don't want to handle your agar dish. First of all, you never want to just open up an agar plate and hold it straight up to the air like this. You're just asking for contamination to fall onto that plate. So don't do that. You also don't want to do the other extreme, which is keeping it completely upside down and trying to inoculate it. You will push your loop into that agar if you do that. What you want to do is open up the plate and hold it at an angle like this. You can get access to the surface. You can get your streaking done. Get the plate closed. You're good to go. The last thing I want to talk about today isn't directly related to aseptic technique, but it's the tools that we have available in addition to what we've talked about already. These are single-use, disposable inoculating loops. They come in a sterile package of about 50. When you open up these packages and you pull out the loop, you can see the handle end is what's going to come out first. So you widen up the mouth of the bag and you carefully pull this out
and you've got a sterile loop at the end. Now, some people don't like these single-use inoculating loops. They are not nearly as flexible as a wire loop. And obviously, they're a single-use plastic item. So you can imagine that when they're being used in a busy microbiology lab, hundreds of them are being used every day and discarded into the landfill. So some people are opposed to their use. It's a problem in all of laboratory work that we use a lot of plastic. We use a lot of single-use plastic disposable materials. And hopefully at some point we will get away from that. But this is a tool that's available to us. And some people really like these because it speeds up their work. This loop allows you to do a single transfer of material and then just toss it into the autoclave bag. You don't need to take the time to put your loop, your metal loop, into the incinerator, wait for it to get hot, then take it out, wait for it to cool. Instead, you just grab a brand new disposable loop. So again, it depends on the lab you're working in, whether or not you'll be using reusable metal inoculating loops or these single-use items. Well, the next item that we can look at is the disposable cotton swab. Those are very useful in the microbiology lab. They come in these sterile packages. There's usually 2 swabs in the package. You can only open these packages at one end. And again, just like with the loops, that allows you to open the package at the end opposite to the swab. So if you carefully manipulate the package here so that the opening is a little bit wider, you can pull the swab out carefully and you've got a nice sterile surface. So you can take culture material up with a cotton swab and use it the way you would use an inoculating loop. When you're done, you can simply toss that swab in the trash. Remember, transfer pipettes also come in these single-use sterile packages. When you open these up, you're going to first see the bulb end. And again, you want to open these carefully and pull them out carefully. But now the end that will touch your liquid broth is sterile. You can pull up a larger volume with these than you could with an inoculating loop, which makes them very nice. Very inexpensive, very widely available, very widely used, these transfer pipettes. Next on the list of tools is one of my favorites. This is called a hockey stick. Now you can buy a reusable glass hockey stick, but this is the disposable variety. Now watch what this stick is going to allow me to do. We've looked at how we streak cells across a petri dish, an agar dish. But sometimes we want to make a smooth coating of cells over the surface of that dish. And that's what the hockey stick does. See, you just swirl it around like the hands of a clock face, and you will spread your cells all over the surface of that plate, nice and evenly. One of my favorite tools in microbiology. At the end of the day, when we've finished our work, it's critical that we know where to dispose of our materials. And of course, when it comes to the materials that touch culture, we need an autoclave bag. This red bag has a biohazard symbol on it, and it clearly says on it, it's an autoclave bag. So this bag is where we put used culture-related materials, the things that have touched living bacterial culture or microbial culture, our agar plates, our cotton swabs that we might have dipped into culture, our disposable transfer pipettes that may have drawn up culture, the cotton swabs, the hockey sticks, all of these things that have touched living cells that we cannot put in the incinerator because they're disposable. We're going to put in the bag. We're going to wrap that bag up with a tie, and that bag is going to go into the autoclave. Now, we haven't talked about the autoclave yet. We haven't talked about the process of autoclaving, but for now, understand that in the autoclave, a very high temperature is reached and a very high pressure is reached. And any cell and any spore that's inside that autoclave is going to be destroyed at that high temperature and pressure combination. Once the material comes out of the autoclave, it is ready to be thrown into the regular trash.
Okay. Any questions about what you saw in that video? Any questions about that process where we flame the test tube or holding the test tube at an angle and putting the caps in your fingers? Sometimes when you see people doing these actual procedures in the lab, sometimes it looks very awkward. You've got an inoculating loop in one hand, you've got two tubes in the other hand, you're trying to hold the caps, it looks very awkward. But all it takes is a little practice to get really good at these things. And unfortunately, we don't have a chance to do that this summer because we're not actually in the lab. But trust me when I say, even though it looks confusing sometimes to have so many items in your hands, with a little practice, you can get very good at it and you can get very quick at it. So I did want to show you something though, while we're on Zoom together. This is a packet of those sterile single-use inoculating loops. I'm going to go ahead and take one of these out because I want to show you what I mean when I talk about tearing agar. This is just a nutrient agar plate that I have here with me today. There's no bacteria growing on this plate, but I put my gloves on anyway. So I'm going to open up this plate. You can see that the agar itself, there's a thin layer of it on the bottom of the plate. Again, it's kind of glistening when you look at it. Remember, agar is the matrix material. It's what makes this stuff solid. It's almost like a gelatin. And just like gelatin, you can add things into agar. So when we talk about an agar plate, we take the agar material and some water, and we add in the ingredients that will help the bacteria grow. We add in the nutrients. We add in whatever else it is we want the bacteria to have. The agar itself is just the matrix. Agar, remember, comes from algae. It comes from certain species of algae. It's what makes up their cell wall in some species. So when you look at it, it looks pretty solid. And when I touch my inoculating loop to the surface, if I'm gentle, I can run my loop all over the surface of this. But if you're not careful, if you dig, you will break the agar. Can you see that? See what I've done? I've lifted it up. It has the consistency of gelatin. It has the consistency of what we might call gelatin or jello. I've got a little piece of it between my fingers and you can see I can squish it. It's solid and we can grow things on the surface of it, but it's not hard. So we have to be gentle. We have to be gentle when we run our loop over it, because if we're not careful, we'll poke through, we'll poke into it and under it. Okay. Some people think that these are harder to use because they don't bend. They're not as flexible. You can see there is some flexibility though. There is some flexibility in it. You just have to practice so you know how much pressure to apply and to not apply when you're using agar. In a broth tube, the cells are growing in the liquid. And remember, it turns the liquid a cloudy color. But on an agar plate, the cells are going to grow in little clumps that we call colonies. And we'll talk more about those in a later lab. So, does anybody have any questions about what we've talked about today? We talked about what aseptic technique is. It's actually a collection of things that we do in order to meet our most important goal. Don't contaminate yourself and don't contaminate your culture. Okay, those are your two big goals. And we have to use techniques in order to prevent those things from happening. We have to handle our culture containers carefully. We have to always be thinking about where contaminants might be coming from. And we have to make sure that when we're finished with our materials, we dispose of them correctly. Anything that touches culture or a diagnostic patient body fluid sample, anything that touches that directly has to go in the autoclave before it gets disposed of, because it may have microbes on it. And those microbes could be pathogenic. We always treat microbes as though they're pathogens, because that way we know we're being as safe as possible. We have specialized bags that we collect contaminated waste in, and those bags go into the autoclave. The autoclave uses heat and pressure to kill cells and spores so that that trash material is no longer contaminated. We also talked about how we have separate waste containers for different kinds of materials. For sharp things, we have contaminated sharps containers. Those have the biohazard symbol on them. We can also collect non-contaminated sharp things in just plain old containers that we can dispose of safely. We put glass into a glass waste box, especially broken glass. And of course, some things we don't have to treat special at all. Our gloves, paper towel, Kim wipes, lens paper, that stuff can just go in the regular trash can. If we're always thinking while we're in the lab about aseptic technique, hopefully we'll keep ourselves safe, we'll keep our colleagues safe, we'll keep the general public safe, and we'll keep the environment safe by not throwing things away that might be harmful for the environment. There are companies that come periodically and collect our hazardous waste containers, our biological hazardous waste, and other things that we can't dispose of ourselves, including our contaminated sharps containers. We have to pay those companies to do that, though. Costs money to get rid of your contaminated waste. Any questions? All right, very good. So remember, if you haven't watched it already, you're going to want to watch the acellular microbes lecture. And you're going to want to take that quiz just as soon as you can. It's not due until Wednesday night, but if you can get it in early, that's best. On Thursday morning, your lecture exam opens. there's a lab homework to do this week, but you cannot do that until after Wednesday's lab. So hold off on that one. The lab practical exam covers up to and including what we did today. That opens up on Thursday morning as well. The lecture exam is due Saturday by midnight. The lab practical exam is due Sunday by midnight. So I recommend doing your lecture exam first. Any questions at all? If you can't think of any now and some come to you later, remember you can message me through Canvas, no problem. And otherwise, I'll see you again on Wednesday morning and we'll do our next lab exercise.
I have a question really quick. The next slide on here says that you can use your notes on the lab practical.
Yeah,
Is that
I
right?
apologize for that. Yeah, that slide was from the lockdown days of the pandemic. I did remove that slide. So if you opened up the slides that are posted right now, that slide's not there anymore. No, you're not allowed to use your notes for exams anymore. Okay. So I do apologize for that. I left that in accidentally and I didn't realize it for a day or two. You're not allowed to use your notes for lecture exams, and you're not allowed to use your notes for these lab practicals, okay? You have to go note free. There are a lot of topics that we've done in lab that are going to be covered on this lab practical, but remember, there's a limited number of questions. So it really is worth your while when you're studying the lab material to think about what I might ask. If you had to pick 2 or 3 questions from each one of those topics, what would you pick? Try to focus on the key things that we talked about in each lab. I will give you this hint though. There are 2 things that I mentioned in labs so far that I told you I want you to be familiar with. When we talked about microscopes, obviously you need to know the pieces and parts of the microscope. I also told you I wanted you to know how to focus a microscope step by step. There's 5 or 6 steps that you follow when you focus a microscope. And I listed those for you in the handout that I gave you. I want you to know those steps. I want you to know how to focus a microscope step by step. So be sure to review that. I also told you I want you to know how to do a gram stain. Step by step. Crystal violet first for 30 seconds. Remember, rinse with water. Grams iodine for 30 seconds, rinse with water. Those are 2 procedures that I want you to be familiar with. I want you to know how to focus a microscope step by step, and I want you to know how to do a gram stain step by step. Remember, there's going to be pictures on the lab practical. So think about the things we've looked at. We looked at cells, right? We looked at cells. We looked at the shapes of cells. We looked at gram status. Purple means gram positive. Pink means gram negative. So if we looked at pictures in lab, be sure to look at those again when you're studying, because you might see them again. All right. If there are no other questions, I'm going to let you go. Enjoy the rest of the day. Get started on all your work. It's a busy week. And I'll see you again Wednesday morning.